Welcome to the HOR Insights YouTube channel.
So today, we're doing a deep dive into a really critical tool for anyone in health economics and outcomes research, the Risk of Bias 2.0 tool. And it all starts with this fundamental principle: garbage in, garbage out. You have to remember, the credibility of any economic model we build or any submission we make to an HTA agency is only as good as the clinical data we feed into it.
All right, here's what we're going to cover. We'll start with the basics. What is risk of bias? Then we'll break down the 5 key domains the tool looks at. After that, we'll walk through the actual assessment process, see how it plays out in real-world HEOR work, and finally, wrap up with why mastering this is so important for your career.
Okay, let's get started. Section 1. What is risk of bias? This is really the foundation of how we appraise evidence.
So what are we actually talking about here? Risk of bias is just a formal way of saying a systematic error, something that causes a study's results to deviate from the actual truth. And here's the crucial part. This is not the same as random error. You can't just get a bigger sample size and hope it goes away. If the error is baked into the study's design, it's there to stay.
And that brings us to ROB 2.0. This is the revised Cochrane tool made specifically for randomized trials. You'll see this everywhere because it's considered the global gold standard. When you're putting together evidence for an HTA submission, using this tool to evaluate the internal validity of the trials is pretty much mandatory.
It's also important to know how we got here. We used to use these old quality scales, like the Jadad score, which would just spit out a single number for a whole study. It was a bit of a blunt instrument. The modern approach with ROB 2.0 is much more sophisticated. Instead of one score, it evaluates bias across different domains, and it does it for a specific outcome, not the whole study. It's also much more objective, using a structured algorithm with what are called signaling questions. So that leads us right into Section 2, the 5 domains of bias. Let's deconstruct the framework and see how it works.
The very first domain looks at bias coming from the randomization process itself. The big question here is, was the way patients were assigned to treatment groups truly random? And, just as importantly, was it unpredictable?
To figure that out, you have to ask 3 key things. First, was the sequence generation really random, or was there a pattern? Second, and this is a big one, was the allocation concealed? Meaning, could the investigators peek and know which group the next patient was going into? That can lead to selection bias. And finally, you look at the results. Are the groups pretty similar at baseline, or are there big imbalances that suggest randomization didn't quite work? Okay, domain 2. This looks at bias that can creep in after randomization, specifically because of deviations from what the trial protocol intended. This is all about blinding and the population you choose for your analysis. Here, you're checking if patients and the study staff were blinded. But for us in HEOR, the most critical piece of this domain is the analysis. Did they use an intention to treat, or ITT, analysis? HTA bodies love ITT because it analyzes everyone based on the group they were originally randomized to, no matter what happened later. This gives the best picture of how a treatment policy would actually work in the real world.
Next up, domain 3, which is all about bias from missing outcome data. You might also hear this called attrition bias. It happens when, for whatever reason, we don't have data for everybody who started the trial.
The key here is to figure out if the missing data is a random problem or a systematic one. Was the amount of missing data pretty much the same in both groups? And were the reasons people dropped out related to the outcome itself? For instance, if way more people in the treatment group dropped out because of side effects, that could seriously bias the results. From an HEOR perspective, a lot of missing data just creates a huge amount of uncertainty when we try to build our economic models.
All right, domain 4 focuses on bias and how the outcome was measured. This is often called detection bias. It's all about whether knowing which treatment a patient received could have influenced how their outcome was recorded. Now, the risk here really depends on the outcome. If you're measuring something totally objective, like whether a patient died, the risk is pretty low, even if the trial isn't blinded. But if the outcome is subjective, think patient-reported pain scores, or an investigator assessing tumor size, the risk of bias is super high if the person doing the measuring knows the patient's treatment. And that brings us to the 5th and final domain, bias in the selection of the reported result. This tackles a sneaky problem called reporting bias, where researchers might only publish the results that look good. So how do you spot this? You have to play detective. You go find the original trial protocol, which is usually registered online, and you compare it to the final published paper.
A huge red flag is if the primary endpoint they focus on in the paper is different from what they said they would measure in the protocol. It's basically a form of cherry-picking the data to tell the best story. Okay, Section 3. Now that we know the 5 domains, let's look at the actual process of doing an assessment, from the signaling questions all the way to an overall judgment. The process is really systematic, which is one of its great strengths. First, for each of those 5 domains, you answer a series of specific signaling questions with answers like yes or no. Then, those answers get plugged into an algorithm that determines a risk level for that domain: low risk, some concerns, or high risk. Finally, based on how all the domains score, you arrive at one overall judgment for that specific outcome.
So what does this look like in the real world? Let's move to Section 4, ROB 2.0 and HEOR practice.
Let's look at 2 scenarios. On one hand, you have an ideal trial. It's double-blind, placebo-controlled, and it's measuring an objective outcome, like stroke incidence. This is very likely going to be judged low risk, and that data is perfect for the main analysis in an economic model. But on the other hand, you might have an open-label trial measuring a subjective outcome like progression-free survival. That has a high potential for bias, and an HTA agency might just reject that data outright or, at the very least, demand some sort of adjustment for the bias.
In our reports, we often show the results like this, in what's called a traffic light plot. It gives reviewers a really quick visual summary of the evidence quality. And notice in Study B, there's only one domain rated as high risk, but that's enough to make the overall judgment for that outcome high risk. That's a key rule of the algorithm. The overall rating is determined by the worst performance.